प्लीज कॉल करो.
हेलो प्रोफेसर राकेश भट्टागर सर.
Hello, Professor.
Hello.
Yes, sir. Sir, are we audible to you?
Yes, you're very much audible. Am I audible?
Yes, sir. It's fine. Okay, sir. We were just checking out because in between, I think we lost the connection. Call karo.
हेलो प्रोफेसर राकेश भट्टागर सर.
Hello. It is.
Yes, I can hear you.
Hello, Professor.
Hello.
Yes, sir. Sir, are we audible to you?
Yes, you're very much audible. Am I audible?
Yes, sir. It's fine. Okay, sir. We were just checking out because in between, I think we lost the connection. Call karo.
हेलो प्रोफेसर राकेश भट्टागर सर.
Hello. It is.
Yes, I can hear you.
Hello, Professor.
Hello.
Yes, sir. Sir, are we audible to you?
Yes, you're very much audible. Am I audible?
Yes, sir. It's fine.
वीडियो कर लो.
ये क्या हो रहा है?
स्टार्ट वीडियो चलता है कि नहीं वीडियो इधर कर लो ना स्टार्ट.
चल नहीं रहा सर ना वो कुछ एरर है नहीं वीडियो कैमरा ऑक्यूपाइड है.
Hello, sir. Good morning, sir. Good morning, Dr. Puri. Are we audible to you?
पार्टिसिपेंट क्लिक कर दे.
Hello, ma'am.
कौन है?
Yes,
Are we audible to you, ma'am?
yes. Can you hear us?
Yes, ma'am. We can
Yes.
hear you very clear. But please put on your video, ma'am. We can't see you.
Video is it's not getting switched on. Off है.
ये जो वीडियो है ना इस पर क्लिक करो. स्टार्ट वीडियो इसको क्लिक करो.
Ma'am, please put on your video if possible.
ऑक्यूपाइड भाई अदर
क्या
तरफ से भी आपको होता है वैसे वीडियो ऑन करना.
नहीं
हमारा कोशिश कान और मंगाना.
Ravinder, tell me now what to do. Ma'am, we all are here actually. So we were waiting for your video to start, and shall we start then?
कैमरा का एक्स्ट्रा कैमरा.
अभी ऑन किया. Okay. Can you see us? Video is on now.
Yes, ma'am. Yes, ma'am. We can see you, ma'am, now. Yes. Yes, ma'am. So shall we start now? Yes, ma'am. We can see you clearly now. Yes, ma'am. Good morning, sir. Good morning, all. So with your kind permission, ma'am, shall we start?
Yes, please.
Thank you, sir. Thank you, ma'am. A very good morning to all. Myself, Dr. Gitika Verma. I'm happy that in spite of COVID-19 situation, we all have gathered here virtually for the 9th Professor R.N. Chakravarti Memorial Oration. So I would request Professor Anuradha Chakravarti, Head of the Department Experimental Medicine and Biotechnology, to officially welcome you all. Ma'am, please.
A very good morning to all of you. Thank you for being here with us today. I, on behalf of Experimental Medicine and Biotechnology, take pride in welcoming you all to this 9th Professor R.N. Chakravarti Memorial Oration. I'd like to express my gratitude towards Professor Rakesh Bhatnagar, Vice Chancellor Banaras Hindu University, the Orator of the Day, Professor G.D. Puri, Officiating Director and Dean Academics, PGI, Professor Gurpreet Singh, Dean Research, PGI, who generously extend their help to make this event a possibility even during this COVID-19 pandemic situation. A special welcome to Professor Chakravarti's family for sharing this memorable moment with us. This year, we are celebrating Silver Jubilee for our Medical Biotechnology course, and we have invited our past students to join this oration. So our heartiest welcome to all of our alumni. So once again, I invite all of you to this knowledgeable and prestigious oration. Thank you.
I now request our Head of the Department, Professor Anuradha Chakrabarti ma'am, to kindly greet Professor G.D. Puri, Officiating Director and Dean Academics, with a flower bouquet. Ma'am, please. clap here since it is happening virtually. I now request Professor Veena Dhawan from our department to kindly present a bouquet to Professor Gurpreet Singh, Dean Research, PGI. Thank you very much, sir. Thank you, ma'am. I now request on behalf of our department, Mr. Shaumik Bhattacharya to kindly present a bouquet to Professor Rakesh Bhatnagar, VC, BHU, Varanasi. Thank you, sir. Thank you. Now, I request Professor G.T. Puri to kindly introduce Professor R.N. Chakravarti and say a few words in his honor and about this oration. Sir, please.
Good morning, everybody. Professor Rakesh Bhatnagar, chief guest for this oration. Professor Gurpreet Singh, our Dean Research. Professor Anuradha Chakravarti, Head of the Department of Experimental Medicine and Biotechnology, PGI Chandigarh. Emeritus professors, faculty colleagues, staff, students, and the family of Professor Chakravarti. It's my pleasure to welcome you all, particularly Professor Rakesh Bhatnagar, Vice Chancellor, Banaras Hindu University, and the orator of the day to this 9th Professor R.N. Chakravarti Memorial Oration. Professor Chakravarti, as you know, was born in Agra in the year 1925, and following graduation from King George Medical College, Lucknow, in 1948, he completed his DTM and honors from the School of Tropical Medicine, Calcutta, in the year 1957. He completed his DPhil Medicine from Calcutta University in the year 1961. Professor Chakravarti was an academician from the core of his heart. He started his career at PGI Chandigarh as a lecturer and rose to become a professor, a post which he continued to occupy with distinction till he retired. He was the founding father of unique and distinguished Department of Experimental Medicine at PGIMER Chandigarh. He was instrumental in developing the Central Animals House facility at PGI, which till date is serving as source of groundbreaking animal research at PGIMER Chandigarh. Subsequently, under his able guidance, an excellent primate facility for the medical research was developed to address various human diseases. Professor Chakravarti was a scientist par excellence. His cutting-edge research in the field of atherosclerosis is acclaimed worldwide. His contribution in research has won citations in various reputed national and international journals. He mentored more than 140 postgraduate theses and 50 PhD students. He was instrumental in initiating the research methodology course for postgraduate students of the institute, and this continues to be conducted till date. He received many honors and awards, including the prestigious B.C. Roy Award in Medical Sciences, as well as a Fellow of Indian National Academy of Science. In reminiscence of his contributions, scientists of repute are invited once a year from various national, international institutes to address the audience and to share their valuable experience in the field of medical research. And the eminent scientist this year and the orator of this oration is Professor Rakesh Bhatnagar, Vice Chancellor, Banaras Hindu University, Varanasi. Welcome, sir. He is very well known name in the field of anthrax vaccine development and recipient of many honors. We are highly obliged to have him here on this occasion and really thank him for sparing his valuable time out of his busy schedule. Topic of the talk is development of recombinant vaccine against anthrax from the clone to clinical trials, which is worth listening, and I am sure all of you would enjoy and cherish it. Once again, I invite all of you to this important and prestigious oration. Thank you.
Such a wonderful introduction to a great legend, Professor R.N. Chakravarti. Now I request our head of the department, Professor Anuradha Chakravarti, to introduce another living legend, Our orator of the day, Professor Rakesh Bhatnagar. Ma'am, please.
So, good morning, Professor Bhatnagar. Professor Bhatnagar, Vice Chancellor, BHU, and our esteemed deans, research and academics, and Professor Emeritus, Senior Professor, colleagues, and students. It's my privilege to introduce the audience, our eminent speaker of the day, Professor Rakesh Bhatnagar, who have very graciously accepted our invitation to deliver the 9th Professor R.N. Chakravarti Memorial Oration today. Professor Rakesh Bhatnagar is a renowned academician cum scientist, presently Vice Chancellor of Banaras Hindu University, Varanasi. He was also the former Vice Chancellor of Kumaon University, Nainital. He did his master's from Kanpur University and PhD from Department of Biochemistry, National Sugar Institute, Kanpur. He has also worked as a scientist in Freiburg University, Germany, and also in the University on Caen in France, besides NIH and US Army Medical Research Institute of Infectious Disease, USA. He also served as an assistant professor at PGIMER from 1983 to 85. He pioneered the teaching of biotechnology at the Jawaharlal Nehru University, New Delhi. He has been awarded the J.C. Bose Fellowship and is an elected fellow of all 3 major science academies of India. He received the Innovation Award from the President of India and as well as the ICMR Award for Outstanding Research in the field of immunology. Professor Bhatnagar is well known for developing recombinant vaccine against anthrax. This technology has been transferred to the industry and the vaccine is undergoing clinical trial. With more than 170 research papers to his credit, his publication has received almost more than 5,000 citations. He has supervised more than 58 PhD students and has been able to generate research funding of more than 35 crore. Professor Bhatnagar research been ranked very high in the anthrax research globally. He is truly a source of inspiration for us. The topic for today's audition is entitled Development of the Recombinant Vaccine Against Anthrax from Clone to Clinical Trial. I am pretty sure that this will be very knowledgeable experience and which we all will be very much benefited from this talk. Thank you.
Thank you very much, ma'am, for such a magnificent introduction. I now invite Professor Rakesh Bhatnagar, our orator of the day, to deliver the 9th Professor R.N. Chakravarti Memorial Oration. Sir, please.
Okay. Good morning, Professor G.D. Puri, the Director of PGI Chandigarh, Professor Gurmeet Singh, Dean of Research, family of Professor R.N. Chakravarti, faculty colleagues, students, and all the people present over there. Let me share with you my pleasant experiences which I had in PGI Chandigarh. As my introduction, it was told by Professor Anuradha Chakravarti that I returned from Germany to join PGI Chandigarh as assistant professor of experimental medicine in 1983. This was my first faculty position, and I was extremely excited to be part of the prestigious institution that is PGI Chandigarh. I still remember Professor R.N. Chakravarti was professor and head of experimental medicine. During those days, we used to call it Department of Experimental Medicine, and later it was expanded to Department of Experimental Medicine and Biotechnology. I remember when I was interviewed by the selection committee of PGI Chandigarh, Professor Chakravarti asked me a very nice question. There were only 3 candidates to be interviewed: myself, my wife, Dr. Nirupama Banerjee, and there was another candidate. At 8 o'clock and used to listen. Chakravarti, 2 times a day, he used to meet all the faculty members and research students in a room called Tea Club, where we used to go and have a cup of tea with him. And he used to have a very informal discussion, and which made us very comfortable that we could interact with him with ease. And any problem we had, we could go to him, and he was like a fatherly figure. Any problem you have, he will help you to solve the problem, and he will give a very, very wise and very, very interesting suggestions, which we as a young faculty members enjoyed a lot. So my tribute goes to Professor Chakravarti. He was extremely good human being. He was a wonderful scientist. very good academicians. I enjoyed his lectures. He was originally as a pathology person, so he could even teach us pathology in a very, very simple language, which we could understand with ease. So I'm very glad that today I got the opportunity to speak as an invited oration lecture in the memory of Professor R.N. Chakravarti. My wife later moved to uh, Institute of Microbial Technology to become a scientist over there. And we stayed in PGI for almost 3 years. And I still remember my daughter went to sector 26 in a Sacred Heart school, which she enjoyed a lot. But somehow, after 3 years, I got an invitation from a medical school of University of Caen in France to spend a sabbatical over there. And we moved to spend a sabbatical over there and then decided to migrate to United States to work in NIH and USAMRIID Frederick. And that is where I started working on the disease called anthrax, which is very important which became very important after 1993 Iraq-Iran war. And further, it gained importance when in 2001, some of the notorious people, they sent anthrax spores in envelopes in Florida, where some of the media persons and some of the congressmen and senators opened those envelopes, and few of them fell sick and 5 of them died. So that became very important to find out how to control this disease and how to develop vaccine against anthrax. I still remember when I went through the literature, uh, once I came back to India to join Jawaharlal Nehru University, West Bengal is still undergoing epidemic against anthrax in the forest. And I had an opportunity to think about developing anthrax vaccine against anthrax vaccine, which will be non-toxic. As you all know, that Pasteur Institute is famous to start work on anthrax, and Pasteur himself developed the very first vaccine against anthrax. So what he simply did, he took the Bacillus anthracis bacteria, which causes anthrax, and grew it at 42.5 degrees centigrade to attenuate the bacteria so that it can become a vaccine. And he did a open trial in the fields to show the first principle of immunology, that if an attenuated bacteria is injected in the animals, and later those animals were challenged with the infectious disease like anthrax, it can give protection. And that was the first very, very toxic vaccine, but that was the first example of developing a vaccine against anthrax. So when I went through all that literature, I saw that the human beings die of anthrax, not because of the infection, because infection can easily be treated by giving antibiotics. However, the anthrax toxin, which has 2 components, I will be sharing my slide and showing you in a while. The lethal toxin is the culprit, which can kill the human beings and the animals. So let me share my slides. And try to tell you with a little visual help. It will be easy for all of you to understand who do not work in the area of infectious diseases. So this is the graphical representation of geographical distribution of anthrax throughout the world. If you see India, India is sporadic. That means sometimes you have epidemics, but after some times it goes away. And some of the blue spots you see, they are endemic in nature, and most of that is in Africa. If you look at this map of India, the dark blue regions where the anthrax is more, but gradually it is moving to the light blue regions, and therefore the infection is increasing by day. This is the, the graphical representation, what are the trend in India. So in 2001, there was a peak, then it is down until 2017. So it sometimes goes up and sometimes goes down. And particularly in Orissa, Jharkhand, West Bengal, and Andhra Pradesh, you get quite often, cases of anthrax. And West Bengal, you see that state had the maximum epidemics of anthrax. Just to look at how the patient of anthrax looks like. Anthrax is of three kinds. The first kind, which is quite common, is cutaneous anthrax. And in cutaneous anthrax, suppose you have a little cut or abrasion in your exposed areas of your body, like wrist, you can see it over here. The spore can get injected in your cut, and that becomes little red and eventually becomes a vesicle and turns into a scar, black scar. You see, it looks terrible. And another kind is of anthrax, which is gastrointestinal anthrax, which is very common in tribal areas in India. Gastrointestinal anthrax is usually one gets by consuming contaminated meat. And as you know, tribal people don't have very good facilities to cook the meat in pressure cookers, etc. And therefore, they sometimes, get an animal who has died of anthrax. They think it's a gift from God, and they take out the meat and they just roast it. Sometimes it is not properly cooked. Some of the anthrax spores get into their gastrointestinal tract and infect the human beings. So there's a lot of pain and fever, bacteremia, and about 50% people die if they are untreated. The third, the most important anthrax is inhalation anthrax. If you inhale anthrax spores, they can be lethal, and about 99% people die who get inhalation anthrax, unless the treatment starts very early with the antibiotics. But the trouble is that you do not see the symptoms in human beings unless it takes about 2 to 5 days where you see the symptoms, but by that time, too much lethal toxin has been accumulated in the body and death is almost guaranteed. And therefore, it is very important to have some very good vaccine which can do the prevention of anthrax. To give you an idea about its infection, Bacillus anthracis is the organism. It's a gram-positive bacteria which causes the disease. It looks like this under microscope. It grows in the chain. It's a gram-positive bacteria. If you look at bacteria more in detail, it harbors two plasmids, PXO1 and PXO2. Both these plasmids are necessary to cause the infection. And if you cure one of these plasmids, the infection will not occur. So these two plasmids are very important, and the plasmid PXO1 codes for three important virulence factors: protective antigen, lethal factor, and edema factor. And the PXO2 codes for polynucleotamic acid, which helps the bacteria to evade the phagocytosis. And all the vaccines which are developed in the whole world are based on protective antigen, and anthrax spores are very stable. They can stay in the environment, in the soil, as long as 120 years, and they can cause infection. So anthrax toxin has three components: protective antigen, lethal factor, and edema factor. Protective antigen, as the name indicates, can give protection to the animals and human beings, Whereas when it is added to lethal toxin, it makes lethal toxin and it can cause death. Protective antigen and edema factor taken together makes edema toxin, which can cause edema in human beings as well as in animals. By the help of these cartoons, I must tell you how the toxin enters the human cells. Protective antigen has a very high affinity receptor on the cell surface of all eukaryotic cells or mammalian cell, all human cells. It can go and bind to a very strongly with the receptor. Then there is a protease sitting on the cell surface. It goes and clips the protective antigen into 63 kilodalton and 20 kilodalton portion. 20 kilodalton portion goes outside and 63 kilodalton creates a space for binding of lethal factor. So lethal factor comes and binds to protective antigen and goes inside the cell, and by receptor mediated endocytosis it goes inside and inactivate MAP kinase enzyme, which eventually causes the cell death or the death of the animal or humans. Edema toxin produces cyclic AMP, which can increase the water level in the tissues and causes the edema. So Bacillus anthracis has another connotation that it can be used as a biological warfare agent. There were stories that Saddam Hussein had a lot of anthrax stored in his kingdom, and it is classified as weapon of mass destruction. The destructive capability of weaponized anthrax is equivalent to a nuclear bomb. And you remember, to create a nuclear bomb, you need a lot of technology. But to create an anthrax bomb, you just have to have little training in microbiology so that you can grow this bacteria. And once the bacteria is grown, you can aerosolize it just like a nebulizer. You put a medicine and it becomes a cloud. And similarly, if you put at Bacillus anthracis, it makes a cloud of Bacillus anthracis in aerosolized form, and it can kill the people. So it's a bioweapon. So there are diagnostic methods are available using CAT scan, smear culture, testing with antibody, and molecular diagnosis by PCR. In Indian scenarios, still, people are using, microscopy and PCR to check the disease. In 2001, let me share the story with you that one of my colleagues from my laboratory in USA, he sent these kinds of envelopes to some people in Florida. In fact, he used to talk about that how to increase the funding in anthrax. And one day while lunchtime, he told us that anthrax funding can be increased just by sending an envelope. And once these envelopes were sent to Florida and some people died, there was an investigation by FBI. FBI came to the institution in Frederick, USAMR at Frederick, where everybody was interviewed because FBI gave this contract to University of Arizona to find out where is the origin of these spores, and they found out that by collecting all the samples from all the laboratories of United States, that the exact genome sequence matched with the genome sequence which was present in some of the strains in US Army Medical Research Institute of Infectious Diseases. So the FBI went over there and tried to interview everyone and tried to find out who is the culprit. And this man, whose name was Bruce Ivins, when he was supposed to be interviewed next day, the previous night he committed suicide, and it was not proved by clinching evidence, but since he committed suicide, people still believe that he would have been the culprit behind sending these envelopes to Florida. So now let us come to how to handle this problem. So the only way to do it, to develop vaccine against anthrax. Till date, the vaccines which are present, see, the worldwide, the veterinary vaccine is nothing but a live spore vaccine which can be injected, and it is very toxic. And second one, Russia is still using spore vaccine for humans. Again, it is very toxic. In UK and USA, they grow Bacillus anthracis in fermenters, and they take the supernatant and precipitate it with aluminum hydroxide and try to use it as a vaccine, which still has some lethal factor, which can cause 5 to 10% residual virulence, and hence there are many side effects which are present in this. But this is the only vaccine which is present and is currently being used by US Army to immunize all their troops, so that when they go to fields to fight, nobody can use anthrax as a bioweapon. Uh, there are some other vaccines which are present in India. Still, we are using spore vaccine, which is an outdated vaccine. So then we thought, why not to develop recombinant vaccine, that means genetically engineered vaccine, which is devoid of all these bad stuff, and it can be safely injected in human beings for giving protection against the disease. So that is what we did. We took out the PX01 plasmid out of Bacillus anthracis. We PCR out the gene. So we made millions of copies of this gene and put it in an expression vector, and cloned it in E. coli, so that it could make a lot of protective antigen. So this is the gel picture which shows that we could make plenty of protective antigen. We purified protective antigen and tried to check whether it is like native antigen or not. So what we tried to do, we tried to see whether it can bind to the receptor of protective antigen or not on macrophage cell line, and the answer was yes, that it could bind equally well with the receptor. So that means we got the right kind of molecule. Then we wanted to see, as I showed you in the cartoon, that this 63 kilodalton molecule was broken down to 63 by a protease. So we wanted to see whether it can be broken down to 63 kilodalton or not, and the answer was yes. Then we wanted to see whether the lethal factor can bind to this proteolytically cleaved molecule or not. The answer was again yes. Then we wanted to see whether it can kill the cell line or not, and the answer was again yes. Whether you take native lethal toxin, or you take uh, uh, my recombinant lethal toxin, it was able to kill the cell line. That means we were working on the right molecule. But finally, the proof of the pudding is to see whether you can really protect the animals against this disease. And it was, as rightly introduced by the earlier speaker, that Professor R.N. Chakravarti, he was pioneer in developing animal models against the disease, and I got interested in testing this vaccine against the uh, animal model. And the easiest animal model is mouse model. Only 5 spores can kill mouse of anthrax. And if you inject the vaccine maybe 3 or 4 weeks before, and then you try to inject the spores of Bacillus anthracis, and if you get the protection, the vaccine is working. And the thumb rule is, take it further. So I'm going to share a little story how I went to the companies. So what happened that since this vaccine was developed in the laboratory conditions, we wanted some company to join hands with us and try to take it further. So what I did, I wrote an email to all big biotech companies who are involved in business of manufacturing vaccines. And all of them replied and they gave encouraging response. But one company decided to send two of their managers within 2 hours of my receiving the email. They came to my office and they said that we are interested in taking this vaccine further. So I said, are you the right person to take the decision on behalf of your company, or somebody else takes the decision? They told me that no, their managing director takes the decision. So I said, then why don't you send your managing director? I only talk to the people who can take decision. So the managing director called me within 5 minutes and asked that they are interested in coming and talking to me. I said, I would like to tell you, if you are really serious about going further, you have to understand two things. I don't talk to the companies free of cost. So my time is precious. So I'm going to charge you 5,000 rupees an hour if you want to talk to me as a company, because you are a profit-making company. I don't make profits. Second, if you like the technology, you have to make some token payment so that we presume that the deal is final. Otherwise, we don't talk to each other. If you agree on these conditions, then you are most welcome to come tomorrow. And the person, he agreed to accept my conditions, and he said that give me 3 hours. 1 hour I want to talk to you and want to understand what this vaccine is. Second hour, I will bring my team of scientists who are involved in vaccine development, in animal efficacy trials, in IPR, patenting, etc., and in human clinical trials, and they are going to talk to you. And if they get convinced, the third hour need to negotiate the price with you. And if we agree on a certain point, I'll be very happy to make a token payment so that the deal is done. So we agreed on that. So the person came and talked to me. And when I explained him with the help of cartoons, because he was a chemistry graduate followed by MBA in marketing, so he said, explain me in simple words. So that is what I did. I explained him how we developed the vaccine and where should it go eventually. So he was very keen and he got interested, and he said that, yes, I would like to call my team of scientists and they are going to talk to you. I said, fine, bring them tomorrow. So next day we sat together, and the team of scientists, I made a proper presentation by PowerPoint. And finally, they also liked the idea. And they said, yes, we would like to take it to the eventual conclusion, whether it can be used for human beings or not. So I said, fine. So then, we sat together, and this managing director asked me that how much money you would like to take to transfer this technology to my company. And even before I talked to him, I asked Biotechnology Consortium of India to evaluate how much money I should charge. And they told me you can ask for 1 crore. So I asked for 1 crore. So when I said we would like to have 1 crore for technology transfer, this managing director immediately agreed. And he said, given. So, I was at a loss. Maybe I asked too little money. He so soon agreed to give us 1 crore. So I tried to increase the price little, and I told him that this 1 crore is for Indian market. And if you want to exploit the international market, the price is 2 crores. So he said, no, I can give you 1.5 crore. I said, why? 1.5, why not 2? He said, I'm not sure whether I'll be able to exploit the international market, but I would like to take both the markets in my hand so that I have exclusive rights. So I said, if that is the case, we can make, there is some echo. Somebody has opened the microphone, it seems. Now it's okay. So he agreed that then I told him that there is another possibility. Let us make two contracts, one for Indian market and one for international market. If you exploit only Indian market, give me 1 crore. If you exploit international market, you can give me 2 crores. He said, no, I want to have only one contract and I'll give you 2 crores. And then he said, Dr. Saab, aur kuch chahiye? So I said, still there is some scope. So I said that, see, you are buying the clone. We are giving you the technology. You are not buying me. So if you need my services, you have to pay consultancy to consult me. So he said, Dr. Saab, how can I do without you? So we will certainly like to hire you as a consultant. So how much will you charge? I said, depends how long do you want to use my services? So he said, at least for 2 years. So I said, okay, you can pay me 1 lakh rupees per month to use me as an consultant. So he said, but remember, this consultancy means you can consult me over the phone, over the email, you can come to my office, have a discussion, but I am not your employees that I'll go and work in your company. So he agreed. But he said, what if something is needed in your company? So I said, if that is the case, then I will send my students and they will help you in your company. But remember, if my students go, you have to pay them. They will not work free of cost. And the price of my student will be 5,000 rupees a day. You can use them as long as you want. And he agreed on that. Then again he said, Doctor Sahab, aur to kuch nahi chahiye. So I thought still there is some scope. So I told him that let us discuss the royalty, and he agreed to give us 5% royalty once it goes to the market. And another thing I talked that I have already filed two Indian patents and 1 US patent and 1 European patent. So I would like, because this is going to protect the market for your company, so you should take future expenses of maintaining and getting these patents. And he agreed to take care of the expenses, although the patents will belong to my name, but we will give him license, but he will take care of all the patenting expenses and maintaining expenses. That he agreed. So on these terms and conditions, we agreed and transferred the technology to the company. I was very glad that it was very well covered by all the media persons, and I was invited to Washington, D.C. in American Society of Cell Biology meeting to talk to the press. And out of 5,000 presentations, this presentation was one of the 10 presentations of things of public health importance, so that you can talk to the press and try to explain them what this vaccine is. So I was very glad that my training in United States, in France, in Germany, and of course in PGI Chandigarh as a young faculty member helped me to bring out this vaccine. So we gave this vaccine to this company, and but before we gave it to the company upscaled it and made it in a near industrial scale, equivalent to 1 million shots per liter compared to the currently vaccine which is available. So therefore, this vaccine is available in near industrial scale. And so in order to explain in a nutshell, what we have done is we have transferred the technology to a company which was already in a business of producing polio vaccine and hepatitis vaccine. Scientists from the company came and got extensive training in JNU. JNU scientists have gone to help Panacea Biotech company to produce 5 batches of this vaccine in their GMP facility. And then this vaccine was given for evaluating preclinical toxicity to a company in Bangalore, Reliance India Corporation, and they found out that this vaccine is without any toxic side effects. So it was uh, given to the further, let us say, development for testing its immunogenicity, and therefore phase 1 and phase 2 open uh, let us say, label randomized placebo control human clinical trials were planned, and until now phase 1 and phase 2 clinical trials have been completed successfully, and they are collecting the data and trying to plan the phase 3 clinical trial. And by the virtue of this, we got several national and international patents to our credit. I do not know how much time do I have.
So we have a shortage
Can
of,
you find?
hello. So we have
Yeah.
shortage of time today actually.
Okay. So what we do is, I think I stop it here. I stop it here, and I only go to my final slide and tell you the conclusions where we are today. All right. Okay. So I skipped, something like 20 different slides. So we have cloned and expressed protective antigen gene. We have bioprocess optimized. We have seen that it is biologically equivalent to the native antigen. The technology was transferred to the company. Phase 1 and phase 2 clinical trials have been completed successfully. This non-toxic vaccine has been given it to the company, and we have also tried to produce DNA vaccine against anthrax. We have found the epitopes which are responsible for giving this kind of protection. We have also cloned this gene in tobacco to develop a vaccine for animals, so that animals may not have this vaccine and therefore it will not be transferred to human beings. Another thing which I would like to share, we have also developed the therapeutic antibody against anthrax. So suppose a person has not taken the vaccine and gets the anthrax disease, the person can be treated by using the therapeutic antibodies along with the antibiotics, and the person can be saved. So, with all these things, we can say that India is in the front line position compared to all developed countries to give this technology uh, to the society to serve human beings. Thank you very much, and let me acknowledge, all my PhD students who are involved in this journey. They work day and night. So quite often I say that they work day and night, and I only talk, which I am talking to you. And I must thank, the funding agencies, Department of Biotechnology, Indian Council of Agricultural Research, Indian Council of Medical Research, CSIR, and Department of Science and Technology, who gave me generous funding all these years for the last 25 years, and my collaborators who helped me at different stages to develop this vaccine. And this is my laboratory in GNU. I would like to give you inside glimpse. It is a BSL-3 laboratory. These days, you are talking a lot about BSL-3 and BSL-2. So just to give you a glimpse, how does it look from inside? And this is how you work inside by wearing your PPEs. And this is my little group currently, and some of them have already gone abroad to work. Thank you very much once again for giving me this opportunity to pay my tribute to Professor R.N. Chakravarti and go back to my uh, beloved institute, PGI Chandigarh, where I started my first faculty position. Thank you very much.
Thank you, sir. Thank you so much. Your journey from clone to clinical trials is really motivating and inspiring, and my heartiest congratulations to you and your team. Now, sir, as per the tradition of our department, a silver plaque is presented to the orator at the end of the oration. Since we are doing it virtually today, so I would request Professor G.T. Puri, Dean Academics, to please kindly receive the silver plaque on behalf of you. Sir, ma'am, please do the honors.
You could just take one. So can you see this?
Thank you. Thank you very much.
Thank you, sir. So we'll send it to you by courier.
Thank you. Thank you. Thank you very much for such a love and affection and honor.
Thank you, sir. Our pleasure, sir. How can I forget Mr. Ranjan Chakravarti, son of our Professor R.N. Chakravarti, attending this event? So I would like to invite him now to say a few words. Sir, please. Sir, Mr. Ranjan Chakravarti, please unmute yourself first.
Yes, can you hear me now?
Yes, sir. Perfect.
Yes. Good afternoon, everyone. I'm Ranjan, the eldest son of Dr. R.N. Chakravarti, and extremely proud and honored to represent all my siblings, Manas, Alok, Sima, and Shikha. I have but one regret on this very happy occasion. Our mother, Mrs. Aarti Chakravarti, who was so delighted when I had first shared with her the news about setting up this institution of R.N. Chakravarti Oration at PGI Chandigarh, would miss the very important event today by just a fortnight as she moved on her final journey on October 16th. But I'm sure she would be extending her good wishes from wherever she is. Ladies and gentlemen, the R.N. Chakravarti Memorial Lecture Series, as I understand, was started with the objective of promoting not only just excellence in the field of medical research, but to recognize and foster a passion among all those who have dedicated their life for furtherance of medical science and that would benefit humankind for many years to come. The endeavor of the series, as we all witnessed, is also to present the most informed opinions on an important issue by inviting an eminent speaker on the occasion to share his knowledge and perspective. We've all been witness to a wonderful, stimulating, and extremely informative lecture presented in such an easy to understand for even somebody like me who's a non-medical person. And I think everyone would have found it very stimulating. Finally, an overwhelming thank you to Professor Jagat Ram, the ex-director of PGI, Professor G.D. Puri, who is the chairman of today's, Professor Arundhati Chakravarti, Anuradha Chakravarti, the HOD of Experimental Medicine and Biotechnology, who was primarily instrumental in setting up this oration, and of course, Dr. N.K. Ganguly, who was supporting this throughout this effort. Thank you once again for inviting us and wishing everybody very well. Thank you.
Thank you, sir. Thank you. Now I request respected Professor Gurpreet Singh, Dean Research, to kindly extend a vote of thanks. Sir, please.
Good morning, everyone. It is customary at the end of such formal occasions to propose a vote of thanks. And it is my pleasure and honor to be doing that today. It's an honor because we are in the presence of Professor Rakesh Bhatnagar, a very eminent scientist, a researcher, and the Vice Chancellor of Banaras Hindu University. It's my pleasure to do this because BHU is my alma mater. And I have lived in the campus from 1961 to 1988. First, because my father was a faculty member there in the IMS. And then I did my medical school training, and then I became a faculty in the IMS before coming to join PGI in 1988. So it's a pleasure for me to propose this vote of thanks. It's been an honor to listen to the journey of the vaccine, which you have just enumerated. I'm sure it will be a source of inspiration to many researchers that things can be done in India also, and then can have worldwide implications. We are very thankful to you, sir, because the COVID pandemic has changed the way we live, and we have all adapted to it, and you have adapted to it by agreeing to do this via teleconferencing. And I must thank the telemedicine department in PGI, which has enabled us to do this and interact with you even at a distance. The presence of Professor Chakravarti's family is always appreciated on this occasion in a way that we remember him and pay our honor to him. And I'm very grateful that Ranjan has taken time out and joined us on this occasion. The other people who have joined us on this conference, faculty members, students, I thank them all for joining in and listening. Thank you once again.
Thank you, sir, for your kind words. And now we have reached the end of this event, so I need permission from the PGI dignitaries to conclude this event. Sir, with your due permission, thank you, sir. So with your due permission, I conclude this oration. Thank you, sir. Thank you, ma'am. Thank you, Professor Bhatnagar, once again for such a motivational speech. And since it's a Corona time, so stay safe, stay healthy, and hope to have such interactions in future. Thank you. Thank you, sir.